Hello to all of you. I am the president of the French Society for Cell and Gene Therapy. And the board and myself are very happy to announce to you today in a series of webinars, a new application for gene therapy of these techniques. I would just like to remind you that we had already 4 webinars on the topic, an introductory one on gene editing and gene therapy, then the application for beta globinopathies, for Fanconi anemia also, and for epithelial cells. So on our website, look at these ones. But today, the one who is going to present is Araksya Izmiryan, and she did her PhD in developmental biology in 2007. And then she started a postdoc fellowship in the lab of Dr. Olivier Danos at Necker Hospital in Paris to develop already a gene editing strategy, but this time a meganuclease-based strategy for also already the purpose of gene correction. And in 2011, she joined the lab of Professor Alain Ovnanian and as a postdoc to develop a gene editing strategy using a meganuclease-based strategy to correct a gene that is very important in the skin, COL7A, and is mutated in recessive dystrophic epidermolysis bullosa. Now, the disease RDEB is a very rare and severe genetic skin disease that is caused by loss of function mutations in this gene and is responsible for blistering of the skin and mucosa. Then she obtained, luckily, a permanent position at Inserm as a research engineer in the same lab, and there she is working on still genetic skin diseases at the Imagine Institute in Paris. She's since 2015 in charge of developing genome editing strategies for epidermolysis bullosa, relevant to transfer to the clinic using relevant strategies to make it clinically relevant to use CRISPR-Cas9 and also the newer base editing approaches for this disease. So she does this also in collaboration with Matthias Titte. And I would like to give you the floor, Araksya, and please let us hear your talk. Thank you.
Thank you, Ez. Thank you, everyone. Just my slide is not okay. Okay. Thank you, Ez. Thank you, SFTCG, to choosing my presentation for oral presentation, and I would like to thanks Matthias Titte for inviting me, you and everyone now I will start my presentation, which will be on the correction of COL7A1 using CRISPR-Cas9 and homologous recombination to treat recessive dystrophic epidermolysis bullosa that I will call RDEB. RDEB is a genetic skin disease, which is genetic skin disease with mucocutaneous blisters and local systemic complications for patients suffering with RDEB. So, they develop blisters, chronic wounds, pseudosyndactyly, esophageal strictures, corneal erosions, anemia, and most severe forms develop a very aggressive squamous cell carcinoma with poor prognosis. And currently, no satisfactory treatment exists. The only ones are symptomatic. So the disease-causing gene was identified in 1992 by the team. The gene is relatively complex, composed of 32 kb genomic sequences, 9 kb of mRNA, 118 exons, which encodes type 7 collagen protein. Type 7 collagen protein is expressed by keratinocytes and fibroblasts. And so far, more than 1,000 mutations have been identified to be disease-causing mutations, with a strong correlation with genotype and phenotype. In relation of the nature of the mutation and the phenotype of the disease. So here I'm showing you the schematic representation of the skin with its 3 layers: epidermis, dermis, and hypodermis. The epidermis provides the waterproof barrier and is composed mainly of keratinocytes. Dermis, which is under epidermis, is composed of connective tissues, and the main cell type is the fibroblast. And finally, hypodermis is composed of fat tissues, and the main cell type is the adipocyte. Type 7 collagen is produced by the keratinocytes of the epidermis and the fibroblasts of the dermis. It is secreted in the extracellular matrix, then assembled into homotrimers, which will form cross-striated centrosymmetric anchoring fibers, which are the key structures of to maintain skin integrity. And they are localized at the dermal-epidermal junction. Here you can see that in the healthy skin, the type 7 collagen expression is nicely localized at the dermal-epidermal junction. And you can also see these anchoring fibers into the lamina densa, which will maintain the integrity of the skin and maintain the adherence. And in contrast of normal healthy skin, in RDEB skin, the expression of this protein is almost or totally absent, and there is also lack of these anchoring fiber structures. So up to date, there are different therapeutic strategies which we are currently developing to treat RDEB. And the most advanced ones are based on the DNA or RNA or protein or cells. So today on my talk, I will focus on gene therapy-based gene correction approach. But I would like also to give you the overview, the other approach, that is the use of cDNA of COL7 by addition into keratinocytes and fibroblasts of the patient cells using lentiviral or retroviral vectors, and very lastly with herpes virus. So my today's talk is based on the genome editing strategy using keratinocytes and fibroblasts, which will be treated with which were treated with CRISPR-Cas9 and donor template to drive the homologous recombination. So my strategy relies on the isolation of primary cells from patients from biopsy, then the maintenance in culture and amplification. These cells later were treated with CRISPR-Cas9 tools and used for making 3D skin grafts that were grafted onto nude mice. So I know that everyone of this audience know very well CRISPR-Cas9 system. However, just a brief overview of this system, which is a dual system composed of gRNA and the Cas9 nuclease. So the gRNA will guide the nuclease into the target sequence, and the Cas9 will drive the DNA double-strand break at the specific loci, and the mutation will be corrected in our case permanently if a donor template is present in the system. However, in contrast of homologous recombination assay, if there is no recombination template, the DNA could be repaired by non-homologous end joining, which is an error-prone assay. So what are the benefits of the strategy using CRISPR-Cas9? So one of the main benefits is that this system allows us to preserve the endogenous transcriptional regulation of type VII collagen. It allows us to accurately correct the disease-causing mutation. It drives a permanent gene expression, and finally, we can correct every mutation that we want to correct in this gene. There are some challenges also to make this system ideal, which are the low efficiency of homologous recombination in terminally differentiated cells. There are still off-target effects which could hamper the expression of other genes. And it is not universal strategy, and we should correct mutations one by one. So here I'm showing you the distribution of 1,000 mutations of COL7A1 between exons, and you can see that here in some exons, example exon 3, exon 73, and exon 80, they are recurrent mutations. And in our strategy, we decided to target mutations which are recurrently present in COL7A1 gene, and we choose the following mutation, which is in exon 80, which is a nonsense mutation, and we have cells at the homozygous state. So our goal of the study was to induce efficiently COL7A1 correction using CRISPR-Cas9 mediated homologous recombination, and to investigate the feasibility and the therapeutic potential of this approach to treat RDEB. So here I just would like to make you a brief overview of my experimental workflow, which consists of firstly to identify guide RNAs which will target efficiently together with Cas9 nuclease the sequences which are mutation specific or in close distance of the mutation to be corrected. Treat these cells with all this combination of guide RNA Cas9 in order to identify the most efficient one, and later to identify the correction rate, efficiency at the DNA, RNA, protein level, and finally making grafts, which will be grafted to the immunodeficient mice. So that's my overview, and I will start to show you the first part of my project, which was to design different guide RNAs and in combination with Cas9 treat keratinocytes and fibroblasts of the patient cells and identify the one which was the most efficient and specific one. Indeed, you can see that among these three guide RNAs that in combination with Cas9 was treated with keratinocytes and fibroblasts, one, the gRNA T1, which is this one, showed very efficient and promising results, estimated to be almost 80% of activity in patient cells. And these results were confirmed by Sanger sequencing and TIDE assay. We also sequenced individual clones of this targeted polyclonal population, and we identify the profile of insertions and deletions in our polyclonal populations, and we see that the deletions or insertions are really ongoing at the target site and not far distances. So we also investigated the specificity of our guide RNA, and we didn't find any off-target using Sanger sequencing and TIDE assay in 10 off-target sequences, which were identified in silico firstly, and then investigated by Sanger sequencing and TIDE. So the next steps were to use this guide RNA in combination of Cas9 to draw homologous recombination and estimate the efficiency of correction. So first of all, we used this guide RNA in patients' primary cells in combination with the repair template. And we sequence it in our cells and find that the correction has happened at the 50% in polyclonal population. Well, these results have been also confirmed by when we performed droplet digital PCR on primary keratinocytes and primary fibroblasts. And finally, we end up with up to 50% of site-specific correction for keratinocytes and up to 46% for fibroblasts, which was quite encouraging results. And that was the very first time that we were able to induce so efficiently the COL7A1 correction without antibiotic selection. So when we sequenced individual clones, we also find the same results at the single clonal level, in which here you can see that many of our clones, exactly 54%, harbor the corrected T nucleotides instead of C, which was the exact proportion already observed by droplet digital PCR. So later we characterized the functionality of our primary cells, keratinocytes, doing bicolony forming efficiency and investigated their clonality after the genetic correction. And we observed that there was no differences between uncorrected and corrected keratinocytes regarding the polyclonal population quantification. And for fibroblasts, we performed growth and confluence monitoring assay. and we didn't observe any differences between these uncorrected and corrected cellular populations, which was very encouraging to start Western blots and grafting assays. So our gene correcting efficiencies were also confirmed by a Western blot assay, as you can see here at the protein level in keratinocytes and fibroblast lysates, we found quite similar efficiencies after Western blots, and the protein expression was also highlighted by immunofluorescence in keratinocytes. As you can see here, in corrected keratinocytes, red cells which were labeled with antibody against collagen 7, while the same cells without correction didn't express any level of type 7 collagen. So these cells we used to produce 3D skin grafts. First of all, we started to load fibroblasts, then we added keratinocytes, and we make the 3D skin cells that we grafted on the immunodeficient mice and keep it up to 10 weeks after grafting. And then we investigated the expression and localization of type 7 collagen at the dermal-epidermal junction as expected. And here you can see that as expected, our type 7 collagen was expressed at the dermal-epidermal junction here in green, while the grafts which were produced with cells without correction didn't express this protein. We also investigated the differentiation profile of our skin grafts, and we didn't find any differences between corrected and uncorrected skin, which means that our skin grafts were functional, and the expressions of type 7 collagen, keratin 5, keratin 10, and loricrin proteins were quite similar. The last experiments that we performed, it was the investigation of ultra-thin skin sections of these grafted mice, and the investigation of electron microscopy. And here we were very happy when we were able to find these anchoring fibers in the lamina densa after 5 weeks or 10 weeks post-grafting, while in skin sections grafted with uncorrected cells, these anchoring fibers were completely absent. So in conclusion, I can tell you that we were able to identify a guide RNA which showed up to 70% cleavage activity in primary RDEB cells without any antibiotic selection. We didn't find any cleavage activity related with off-target sites. We confirmed the efficacy of recombination up to 56 and 41 in keratinocytes and fibroblasts respectively at the DNA and protein levels. We were able to to use these cells to produce grafts and graft to the immunodeficient mice, in which the functionality of collagen 7 was analyzed and proven through the presence of anchoring fibers. And finally, we hope that this strategy could pave the way for genome editing for RDEB in general. And with that slide, I would like to thank all of you for your presence, and my team, Camille, Sonia, Olivier, who did a lot for this project, all the other members, our collaborators, our founders, and you for the attention. And here is my last slide that maybe S would like to comment. Thank you.
Thank you very much for this brilliant talk. I just wanted to tell to the audience that, of course, the SFTCG is co-organizing this year the ESCGT, so the European Gene and Cell Therapy meeting in Brussels. So we encourage you all to submit abstracts and to go. Brussels is not so far from France, so you should be able to attend. We will go over a couple of questions that I would like to ask Araksya. Can you stop sharing now?
Yes.
Thank you. Perfect. So Araksya has a bit of problem with the camera. She's cut off, but you were right in the middle. No problem. I have a general question, because you said that there are 1,000 different or a lot of different mutations in the COL7A1 gene. So by gene editing will always remain a personalized medicine in that case, right?
Yes.
But if you use a gene addition strategy, my first question is, how can you do that? Because the gene is enormous. And then you would be able, I suppose, to tackle all the different mutations.
Well, first of all, thank you for the question. I will start with the genome editing purpose, and then I will switch on gene addition. So in our strategy, we will try to target the recurrent mutations, which are not so many, but they are so recurrent that it will allow to at least target from 10 to 30% of population harboring these mutations. We know that there is one mutation in Exon 3, which is very frequent in Central European populations, and currently we are working on it. And the Exon 80 harboring mutations are very frequent in people with Spanish background, and it will address at least for 40% of people with RDEB. So our purpose is now to develop personalized medicine approaches on especially on recurrent mutations. But if I switch on the gene addition, so 9kb mRNA cDNA is already cloned in a retrovirus and lentivirus. The lentiviral purpose is developed by Matthias and Alan in our lab. And there are also teams from US which cloned successfully the 9kb cDNA in a retrovirus, and they can target very efficiently the cells, and there are already ongoing clinical trials with the retrovirus. At least 8 RDEB patients have been treated, and very lastly, this 9kb cDNA were cloned into herpes virus, and they developed also in local approach based on local injections. So it's quite promising, but still the use of retrovirus using aleatory integration is an issue. So the genome editing is something which is very personalized, but at least we will be able to permanently correct and restore the the function of the type 7 collagen.
Yes, I get it.
There are strategies which are developing in parallel. All are good.
Yeah, sure. I understand there's a big difference between the gene edition, where you integrate somewhere in the genome of the skin cells your sequence, and you don't know where you're going to land, and if you're going to, maybe it's not been shown up to now, activate an oncogene or a proto-oncogene, while the gene editing strategy will on-site correct the endogenous gene. So if I understood it well, they are not only proposing ex vivo correction of the skin cells and then making of the skin to transplant the skin, but they also directly in the blistered areas inject vectors.
That's the purpose of the herpes virus, the local injections.
Okay. That is a good, a nice strategy to avoid all the different steps,
of course,
before
the use of maybe keratinocytes which are not which are not so stem.
Okay. And just for the audience also to know, because everybody has a skin, but how long does it take to make a graft for implanting in a person? Do you have an idea?
Well, I think the best people,
or already for a mice, I don't know.
Yes, I think for mice it's 3 weeks, and it's a very small centimeter square area. I think it when we use for human, we will use more cells, but it will not go more than 3 weeks in culture. But when it is grafted, so if the grafted product, if the cells used for graft are healthy and containing holoclonal populations, it should maintain. So with in the study with Americans that they conducted for 8 RDEB patients, they make follow-ups till 1 year after grafting, and but they see that after 1 year, the type 7 collagen expression was progressively decreasing.
Okay.
Because the point is also that maybe we use the cells, but these cells will not continue to proliferate more than I don't know, more than one year.
Because you lose a bit their phenotype.
That was the purpose. That was the purpose of the study, because after 1 year, they were losing the collagen 7 expression. But the use of local injections maybe is the key for success now at this point. Because even this, the half-life of type 7 collagen will be 3 months or maybe more, maybe less, at least with local injections, which are not so intrusive. So we can maintain somehow the wound healing process ongoing.
Okay. Okay. Then I have a small question on the technical question. You used the I think oligo sequences for your as template for your DNA repair, right?
Yes.
And are they somehow, because SSODN, are they modified or stabilized or?
They are basically stabilized with 3' and 5' modifications, but
Okay.
there is nothing special on it.
Okay.
It's the basic stabilized oligos from IDT.
Okay. And one small question still. I saw when you have 56%, for example, correction at the right site in your gene, that when you apply the CRISPR-Cas9 gene editing, you have on top of that insertions or deletions. Right?
Do you mean my single clonal
yes, when you looked at the different clones, you had different.
Yeah. So by droplet PCR, I find up to 50% of correction in the polyclonal population, but it was also confirmed by these single clones, which contain the reparation of the right nucleotide, but still they contain some insertions or deletion at the target site. Hence,
is that problematic? That's my question.
Well, could be problematic if the target site is exonic.
Yes.
But in my case, I put it into intron and not in regulatory.
That's what I thought, but it was not clear.
So it could be problematic because somehow we were not able to do homologous recombination 100% accurate. So that's why it's it's challenging. But if we are able to play with these distances, putting the target site in introns, maybe for me it was not hampering. But for this mutation,
for sure. Then I mean, in the end,
yes,
the cDNA, you will have no residual.
Yes,
of these.
but now there are also base editors and prime editors which will not make cuts. So we can also play with these new tools to avoid these non-intended, non-homologous and joining pattern with in combination with homologous recombination.
Okay, well, I think we will leave it at that, Araksya. Thank you very, very much for your answering my questions and for your presentation once again.
I was happy. Thank you for inviting me.
No problem. So I will say hello, bye to everybody, and really look at the other webinars. They are really interesting on the site, and don't forget the next ESCGT meeting in Brussels. I hope to see you all there. Thank you.
Thank you.